Hey everyone, in this video lecture, we are going to be talking about the steps involved in protein synthesis. We're going to talk about the step 1 of making proteins called transcription. So to recap a little bit about protein synthesis, the overall process of making proteins, it is a two-step process. So we know our DNA acts as the instructions for how to make a protein, but the problem is we can't directly use the DNA to assemble proteins. So step 1 is we're going to take all the DNA that you have in a cell, and we're going to take one little section of it at a time. That section is going to make one single protein. And so we're going to copy that one section of DNA into an RNA message. And throughout this video, we're going to see why that is. And then once we have that RNA message, we then go through the next step in our cells where we take that RNA message and translate it into a protein. Or really, we're making a sequence of amino acids. We're putting together the amino acids in order to then make a functional protein. So to do this, we need this process of transcription, and we're taking that DNA and we're making a complementary strand of RNA. So complementary meaning if I have the letter A in my DNA, I'm going to pair it with its complement, which is a T. However, we're making RNA, and RNA does not have T's, it has U's in the form of uracil. So uracil, or U, would be the first nucleotide of the RNA message. If my next letter is a C in my DNA, then the next complementary letter in my RNA would be G. So it's a complement. It's not like if the DNA is A, then my RNA is an A. We're making complements, meaning we use the base pairing rules. So thinking about where DNA lives inside of the cell, where must transcription take place? If DNA is stuck inside of the nucleus, well, that means transcription has to be occurring in the nucleus. The problem is that DNA is the instructions to make proteins, but where we make proteins is not inside the nucleus. The factories that make proteins in our cells are ribosomes. Ribosomes are not found inside the nucleus. They're found out in the cytoplasm, or they're found on the endoplasmic reticulum. So the reason DNA has to be copied into RNA is that DNA cannot leave the nucleus. It is too big of a molecule to leave the nucleus. So instead, if we can copy little sections at a time into these RNA messages, the RNA message is small enough that it can leave the nucleus and get to a ribosome outside of the nucleus. Okay. So the fact that DNA is in the nucleus means transcription is going to occur there. And the reason we have to copy that DNA into RNA is just the DNA is way too big to leave the nucleus in order to make proteins. So we have to use RNA as kind of that intermediate molecule to make proteins. So again, just to review the differences between DNA and RNA, the big difference here is those nitrogenous bases of the nucleotides between DNA and RNA. DNA has T's or thymine, whereas RNA does not. It has U. So when we're trying to figure out those complementary pairs, we need to remember if the DNA has an A, then it's pairing with a U in RNA. If there's a T in the DNA, it would still end up pairing with an A. So we'll see that in a minute. So the way transcription occurs, or the machinery necessary for transcription to occur, we need an enzyme. Because remember, enzymes are special proteins that do very specific jobs inside of cells that would not happen without them. So RNA polymerase is an enzyme that is going to help build RNA molecules using DNA. And we're going to see how it does this, how RNA polymerase is able to take the DNA and actually create this RNA message. So step 1, in order for transcription to occur, is we need to initiate it. So step 1 is called initiation. And what happens is your RNA polymerase enzyme is going to attach itself onto the DNA at a sequence within the DNA called a promoter region. So it's a sequence of letters or a sequence of nucleotides in the DNA that the polymerase recognizes. So once it sees it, recognizes it, it binds to it. It's like, think of like magnets are attracted to each other. It's kind of like this sequence of DNA nucleotides attracts this RNA polymerase, it binds to it. So once RNA polymerase attaches to the DNA, it causes the two strands of DNA to split apart. It opens up that DNA strand. And so this promoter sequence is always going to be found near the beginning of a gene, genes being sections of the DNA that make a specific protein. So that little promoter region will always signal the start of where you want transcription to occur in a section of DNA. So once that polymerase is attached to the DNA and the DNA splits open, now what the polymerase enzyme is going to do is it's basically, in a way, going to read each DNA nucleotide and add a complementary RNA nucleotide. So you have your top strand of DNA is not being used. We call that the coding strand because it's going to have the same code or the same nucleotides that your RNA will. And then the strand actually being used to build the RNA gets called the template strand. And so polymerase is going to literally go nucleotide by nucleotide and add and bind the complementary RNA nucleotides to the DNA. And then as it continues kind of plugging along the DNA, the RNA message will then kind of be spit out of the RNA polymerase enzyme. And so that will continue until the entire section of DNA needs to be transcribed. And then once there's kind of an ending point, we reach what we call termination, the last main step of transcription. So there's what we call a terminator sequence within your DNA that tells RNA polymerase it's time to stop making the RNA message. So terminators are sequences within your DNA that once the RNA polymerase reaches it, basically, RNA polymerase is going to fall off the DNA. It will release that mRNA message, and transcription is over. And so once we have that little piece of mRNA, transcription is done. So now we have copied DNA into this RNA message. But before that RNA can then go on to the next step of being translated into a protein, that RNA needs to be modified. And so in RNA, or actually in your DNA, there are sections of DNA that are kind of useless. There's parts of your DNA that are useful for making proteins and parts that are not necessary. So the sections of your DNA called introns, these are just sections of nucleotides that don't do anything. They don't actually code for amino acids, so they're not needed to make proteins. So what has to happen is these introns, or sections of the RNA, really the DNA, that are not necessary for making proteins, get snipped out. So then the parts that are used, called exons, they're going to remain in the RNA message. And RNA is a very unstable molecule, so we also modify it by adding caps to the front and kind of the back of the RNA message to make sure that it's protected as it's moving from the nucleus to the ribosome. So at the front, there's a little cap, and at the end, we call it a poly-A tail, meaning they add a bunch of A nucleotides at the end to protect it, so there's no issues transferring that message from nucleus to ribosome. So pause the video here and do a little bit of practice where you have a section of DNA and then we want to transcribe it into mRNA. So for example, if the first three letters of DNA of that template strand are GAG, the complementary mRNA strand would be CUC. Because remember, there are no T's in RNA, so wherever there's an A in DNA, it would pair with a U in RNA. So continue on and complete this practice problem.